There we are. Well, buenos tardes, everyone. And thank you ever so much for inviting me to give this talk. It's a great opportunity to talk about what I think is an important topic, but it's an often forgotten milestone in the history of visual optics. The new work in this presentation was published recently in the Journal of the History of Neuroscience. And so the story that I'm going to tell you today would have remained largely untold without the expert translation of many books and papers in the 19th century German literature. Unfortunately, I do not speak or read German, so I depended deeply upon my co-authors, Katharina Lehner and my son Cameron, who is fluent in German. Now, everyone knows, of course, in the modern world that rods and cones are the elements of the retina that respond to light, and they initiate the visual process. Retinal diseases that affect rods and cones are common, and their devastating consequence to vision of people are understood by everyone. So I think it's important for we scientists and clinicians to know who discovered that rods and cones are the eye's photoreceptors, and how did they do it, by what means. Before focusing our attention on this historical question, however, I think it will help to recall how we came to understand the eye more generally as a biological optical imaging system. Well, image formation by the eye was already established in the 17th century. After a lot of confusion and wild speculations, it was Kepler who really established a definitive idea. Probably because he was familiar with the telescope optics, he was the first to argue on optical grounds that vision starts with the formation of an inverted retinal image cast upon by the eye's optical system, the cornea and the lens. There's a direct quotation from his book on astronomical optics. He said, "I say that vision occurs when the image of the whole hemisphere of the world, which is in front of the eye, is formed upon the reddish-white concave surface of the retina." I leave it to natural philosophers to discuss how this image is put together by the spiritual principles of vision residing in the retina and the nerves. Well, you can tell from the form of the language here, he wasn't as comfortable with psychology and perception as he was with optics. And in fact, Kepler had a very long wait. It would be another 250 years before those natural philosophers could have satisfied his curiosity. Now, contemporary with Kepler, we have Christoph Scheiner, who in the year 1619 confirmed Kepler's assertion by direct observation of the formation of the retinal image. And he did it very simply using a sheep's eye. He went to the abattoir, got a sheep's eye, and I dissected it from the backside to provide a viewing window onto the retina, viewing from behind. And there you can see the image of the world, inverted just as Kepler had predicted. Now, Descartes is responsible for this lovely little drawing over here of Scheiner looking at the back of the sheep's eye. I think he was a big fan, no, Descartes. He was a big fan of Scheiner. He used this illustration many times in his book about dioptrics. And Descartes, in fact, quantified and made it much more optical looking kind of diagram here when he created a schematic model of the eye to explain how the topographic mapping of the visual world onto the retinal surface occurs by simple ray tracing. Yeah, click, there it goes. Okay. Now, in this same era, we have the first Spanish book on visual optics, which I've learned recently was published in 1623 by Benito Daza de Valdez, if I pronounce that correctly. And according to Google translation tools, this title means spectacles for all kinds of visions, in which it is taught how to know the degree that each one lacks in his vision and those who have any desires. It was dedicated in the year 1623 to the Lady of Cordova. A very famous man, I understand. The optical center in Madrid is named after this gentleman. Now, despite those 17th century advances in the understanding of the eye as an optical imaging system, the required visual mechanism that was needed to sense the retinal image remained unknown for another 2.5 centuries. It's amazing it took so long, until the middle of the 19th century. And the two men who figured it out are shown here. One, Carl Bergmann on the left, Heinrich Müller on the right. These are the protagonists of my story today. They are the men who discovered that rods and cones are the eye's photoreceptors. Now, it's rather symbolic in this slide that the two scientists are not looking at each other. And in fact, I suppose if they had guns in their hands, they might have had a duel at this point. They were not the best of friends. They were not on good terms. Müller had a very long career with many publications in retinal anatomy and is justifiably famous, very well-known anatomist. But he was highly critical of Bergmann's results and experiments, as you'll learn as I carry on with this presentation. In fact, Bergmann published only 2 papers in vision before leaving the field for other topics, and quite possibly because of Müller's harsh criticism of his work. So sadly, Bergmann's contribution has been almost completely forgotten. Now, before telling you the 19th century story about how the eye it was discovered that the eye senses the retinal image with rods and cones, I'm going to give you a brief review of retinal anatomy as presently understood, so that it will help to reveal to you by comparison the state of ignorance that existed prior to the pivotal year of 1854, when Bergmann and Müller independently concluded, using very different methods, that rods and cones are the eye's photoreceptors. So for anybody in the audience listening now who is not familiar with ocular anatomy, this schematic drawing that you see here represents a horizontal cross-section of a human eye. The eye's optical system consists of 2 lenses. One is the cornea, which has the greater optical power, and it is fixed in its optical power. Now, the internal lens here is flexible. It changes shape, which changes its focusing power when viewing distance to objects changes. Now, together, this 2-lens system casts an image of the external world onto the retinal surface. Visual acuity is greatest in the foveal region. So when we want to see a detailed feature of an object, we rotate our eyes in order to place the image of that object onto the fovea. Now, a highly magnified view of a cross-section of the retina here is shown at the top. There are 3 main layers of retinal neurons. It's a 3-neuron chain from image to neural response. On the inner surface of the eye lies the retinal ganglion cells. These are the output neurons of the eye, and every cell produces a long axon going out along the surface of the retina, and down in this diagram has to travel all the way to the optic nerve and then exits the nerve and goes all the way to the brain. And it's a long trip. Comparatively speaking, it's about 10 centimeters or so to get to the brain. And this retinal thickness is on the order of about a 0.3 millimeters in thickness. So it's a long trip in terms of physiological distances, anatomical distances. Now, on the other side of this retina, on the outer surface, we have the rods and the cones. And in between, there's a layer of interneurons that relay messages from the photoreceptors through the relay neurons to the ganglion cells and then out to the brain. Now, the eye also has important topographical features that are best observed in plan view, as seen from, as if you were in the center of the globe looking down onto the surface of the retina. Let me explain each of these 4 diagrams. The first diagram shows the eye as a hemisected view, and it establishes kind of a terminology convention for 2 important directions. One is the radial direction. That's these little blue lines. These are lines through the center of the globe, and they pass perpendicular to the surface of the retina. They are to be compared with the yellow lines, which are in the surface of the retina, and they are running laterally, parallel to the surface of the retina. Now, part of the big argument between Müller and Bergmann has to do with whether certain features of the eye are radial or are they lateral. And they both had very strong opinions on that topic. And it was a critical point to decide. And the second picture in here, in part B, this is a picture of, or drawing really, of the retinal vasculature. See the retinal blood vessels, the arteries and the veins, they emerge just from the optic nerve head. So that one hole in the back of the eye is a conduit for both the nerve fibers of the optic nerve and for the blood vessels. And these blood vessels spread out across the whole surface of the retina. And there are actually many more vessels that you can't see because they're tiny little capillaries, but they cover the entire surface except for one special place, and that's the fovea. Now you might think that's kind of interesting that the blood vessels don't enter the foveal region. Now it's pretty strange when you think about it because the fovea is a region of highest acuity and best vision. How can it be surviving if it's not perfused by blood vessels? And the answer is that its blood vessel, its supply of nutrients comes from a different blood vessel supply. It comes from the choroid, which is behind the retina, and the nutrients are transported across the retinal epithelium to nourish the fovea. The bottom diagram in part C down here shows another kind of a radial pattern over the surface of the retina. Again, starting at the optic nerve head on the left, streaming out across the surface of the retina. These are the fibers of the optic nerve. So, of course, the optic nerve fiber starts with the ganglion cell. So imagine a ganglion cell position where my little pointer is located. It generates an axon. That axon has to find its way all the way to the optic nerve head, but it cannot go through the fovea. It's one of the rules of development of the primate retina. Thou shalt not pass through the fovea. The fovea is a special place. Axons do not go that way. And again, that was a very important point to be resolved. Bergmann understood it. Müller disagreed. And that was part of their contention. And finally, we have on the right-hand side another plan view diagram. And this is positioned right at the edge of the fovea over here, where you're just starting to leave the fovea and go into the peripheral retina. And on this diagram, with all these little black spots, these are the nuclei of photoreceptors. On the right-hand side are the nuclei of the interneurons they have to connect to. And so what you have are long fibers running from every photoreceptor as a long axon that goes this whole distance and ends up making synapse with its interneuron. And in order to get over there, it has to run along the surface of the retina. So they are actually running parallel. These are the laterally running fibers. And again, it was a bone of contention between our 2 protagonists. Now, in the modern world, we're really spoiled, really. We have such fabulous anatomical methods available that are based on histochemical techniques and optical methods. We can get beautiful views, even of the living human eye. And here's an example of it's almost a color-coded picture of a cross-section of the human retina, not on a living eye, but it's been treated with histochemical methods, starting at the optic nerve head on the left, a beautiful panoramic view of the human retina spinning all the way across through the foveal pit and then out to the other side. And what I want you to notice is that the layers are beautifully represented by the different colors, and the retina is very thick in the periphery. But see what happens when it gets closer and closer to the fovea. When you get to the fovea, the retina becomes thin. And in fact, it is so thin that the only elements that remain are the photoreceptors. These are the blues and the greens at the bottom. Again, a point of contention between our two protagonists. The second image here, this one in black and white, this is the spectral domain of image taken of a living human eye using optical coherence tomography, modern optical technique. It's running through the optic nerve fiber, optic nerve head on the left, in the same way. Now, what I specifically want you to notice is that this little dot right there, and that little white dot there, and this little white dot over here, these are blood vessels, and they're running parallel to the surface of the retina, so they just appear as little dots. But look what's below them. There's a black line extending down. That's the shadow created by the imaging light that's used to collect this OCT profile. So there's a shadow being cast by the opaque blood vessels, and that was that was the feature of the retina that Müller depended upon in order to make his discovery. And then finally, down here's some more beautiful color coding of all the retinal line elements right near the foveal pit, and you can see the ganglion cell layers getting thinner and thinner and thinner, the foveal pit being over here on the left, and the internuclear layers getting thin. Everything's thinning out except the photoreceptors. They remain positioned there. Now, the final image to call your attention to is this beautiful viewing of just the photoreceptors. And you can see the rods and the cones here, the outer segments where light is absorbed, the inner segments that collect light and funnel it by waveguide action into the outer, to the pigment in the outer layers. But notice what happens to each axon. Here's a beautiful one right here. That cone sends out an axon. It's got to turn right and head off and run almost parallel to the surface of the retina to find its inner neuron to make synaptic contact. Bergmann observed this for the first time and commented on it in his paper, and Müller denied it quite emphatically. He said it doesn't happen. Another bone of contention between those two gentlemen. Okay, so that's enough anatomy. You now see all the things to pay attention to when we discuss what happened back in that 19th century. So let's take a time ride now, a ride on a time machine, and go back first stop the 1900. At the end of the 19th century, the definitive work by the great Spanish retinal anatomist Ramon y Cajal in Madrid had firmly established what he called the neuron doctrine. And that was that the retina and the central nervous system in all vertebrate animals, including man, is an interconnected network of individual neurons. And that key idea, which we may now take for granted, of course that's the way it is, but prior to Cajal and the work in the 19th century, that was not appreciated. It didn't realize that these were individual neurons. The photoreceptors at the top, inner neurons, and then the ganglion cells. A 3-neuron chain to get that retinal image sensed and sent off as a neural image to the brain. Now let's take the time machine back another 100 years, go back to 1800. What is a life like then? Well, at the beginning of the 19th century, very little was known about the internal structure of the retina, primarily because poor optical quality of microscopes. It was noted that the spreading of the optic nerve fibers had been seen through by dissection, really, that these optic nerve fibers spread across the inner retinal surface, and that suggested that these fibers, or the terminal ganglia, that they at the end of that fiber, they were responsible for sensing the retinal image formed on the inner surface of the retina. See, in this diagram on the left from 1849, the English neuroanatomist Baumann shows light coming in from the bottom, because in this diagram, the light's coming up, and the optic nerve fibers are at the very first thing, and these are the ganglia where they terminate, thought to be terminating there. Not sure what a ganglia is. It wasn't really a cell. We didn't understand that yet, but it was some kind of thickening of the optic nerve fiber. And what people naturally thought was that this is where the retinal image is formed and sensed by these ganglia. And all the rest of this material up there, they had absolutely no idea what it was good for. It was just tissue, didn't know what. Well, moving forward in time, scientific histology developed rapidly in the 19th century due to improved microscopes. The optical quality was improving rapidly, and there were better techniques being developed for fixing and mounting biological specimens, because in those early days, the anatomists were working with fresh tissue. It was notoriously difficult to work with. It was very thin and fragile, easily broken, impossible to work with. It really had to be fixed and made hard to be able to really make progress in this field. So by mid-century then, in 1849, Baumann's anatomical drawings suggested that the retina was kind of like sedimentary soil, rocks and soil of different grades. We just had layers of tissue. Some layers have coarse elements, some had fine elements, and they had names such as the granular layer. Who knows what the granular layer is? Turns out those were the photoreceptor nuclei, but nobody knew that then. And the bacillary layer, bacillary just means rods. And so up here is the bacillary layer, the rods and bulbs, later called cones. And these turn out to be the inner segments and outer segments of photoreceptors, but that was not appreciated at that time. There was essentially no sense at all that the retina is a network of interconnected cells. That didn't happen until really in Cajal's time, the second half of the 19th century. Now, we're going to park our time machine here, get out of the machine, have a bit of a walk around. The year is 1854. This was the pivotal moment in the history of vision science, when two independent arguments, one physiological and the other anatomical, indicated that rods and cones are the eye's light detectors. Now, the physiological argument was by Müller, and it was based on his earlier discovery in 1851, just a couple of years earlier. He discovered radial fibers traversing the full thickness of the vertebrate retina. If you look back at Kohler's diagram, there's no sense of any kind of fiber spanning this space. It's just tissue, no fibers. But Müller said, no. There are fibers running from the inner to the outer surface. And so he discovered that, and then based on that discovery, Müller and Kolliker, his friend, another anatomist, independently suggested that these radial fibers are actually an extension of the optic nerve fiber, which passes up through the ganglion body and terminates as rods and cones in the outer layer of the retina. So back up again, what he's imagining is these radial fibers run from this optic nerve fiber through the ganglia and make their way up here and terminate at the rods and the cones of the bacillary layer. And that is what he's going to say. He speculated that is where the light is sensed and is directly output, skipping all this intermediate tissue. It has no relevance. It's just physical support structure. And that's how light is sensed. That was his hypothesis. Now, the implication one is that light has to pass through the retina before it is detected by the rods in the cone tips of optic nerve fibers. And having created this hypothesis, he set it to test. As a good scientist, he said, I'm going to test my hypothesis. And in the year 1854, he did the experiment and confirmed his hypothesis was true using Purkinje's 1819 method of visualizing the retinal vasculature anatopically. By that means the object of an anatomic percept is a perception of an object that is inside the eye, and those objects were the blood vessels. And again, as I mentioned earlier, it's all based on the shadows cast by the blood vessels. So that was the first argument. The second argument was anatomical, and this was by Bergmann, same year, 1854. Now, Bergmann had access to a human retina. See, Müller had never studied a human retina. He had studied many different kinds of animal retinas, but he just didn't have access to a human. But Bergmann did. He had the eye of a prisoner, a criminal who had been executed, and he was able to get the eyes and fresh, dissect them, and observe them under his microscope. And his key observation was that the central macular region, which is where the yellow spot is, that's where the fovea is, there's a little marker there, some yellow pigment. And then that area, it contains the specialized area, the fovea. He called it the fovea centralis, the central fovea, which was previously thought to actually be a hole in the retina. He said it's not a hole. He said this fovea centralis, it may be thinner than the surrounding region, and the reason it's thinner because the optic nerve fibers and the other layers of the inner retina have been displaced laterally. That tissue has been moved out of the foveal pit, put it to the side, a bit like, in my mind, it's like digging a hole in the garden. You have to pull dirt out and you put it next to the hole there. It's just, the dirt doesn't go away, it just gets piled up on the side. And that's the kind of picture that Bergmann described in his paper. So in this area of acute vision, he said, there are only elements there are rods and cones. This is the area of best vision. The only elements present are rods and cones. Therefore, they must be the light-sensitive elements of the retina. There's no way around it. It's a conclusive argument. So for anybody who needs to leave the Zoom meeting early, you can go now. This is the answer to the question I posed. These are the two men who had discovered rods and cones by very different methods. And what I'm going to tell you next is how they did it. So stick around. So let's look at Müller's experiment first. It's actually quite a famous experiment. You'll find it described in many textbooks. And very recently, Werner and colleagues actually gave a very nice detailed description of those experiments, put them into context, and it was published in the Journal of the History of Neurosciences. So Müller presented in the year 1854 evidence supporting his earlier conjecture that light is being detected by the bacillary layer of rods and cones, which he thought were the terminal endings of optic nerve fibers. Now, he achieved this fundamental discovery using ancient principles of the sundial. Now, the sundial has been around for a long time. I mean, the earliest known examples of a sundial go back to Egypt, Valley of the Kings, 1500 years BC. You know, this is 3,000 years before Müller was born. It's a very old concept. And there are two key principles of the sundial clock that Müller made use of. The first is that light casts shadows on the opposite side of an opaque object. So here we actually have a modern human sundial. Person standing here, the sun shines on, and the shadow is cast on the opposite side of the sun. And the second principle to know about is essentially motion parallax. That is that the shadow is going to move when the light source moves. And that's why the sundial can be a clock. When the sun moves, the shadow moves, and you can tell the time using the markers on the ground. Well, those are the two principles he's used, and here's how it worked. Purkinje had already discovered, Purkinje being the great, the Czech physiologist, he, in his PhD thesis, by the way, one page of his PhD thesis, he described what has been forever known as the Purkinje's tree, which was an anatomic visualization of his own blood vessels. And here's his drawing of his own blood vessels. Optic nerve head, here are the major vessels coming out. And right there, that little circle is the place where he said there were no blood vessels there. And it turns out that's the fovea. Now, well, that's the drawing, and Müller says, I'm going to do the same experiment, set up the optical system such that I can have a little candle down here and let the light from the candle go into the eye, and it's going to hit a blood vessel, and it's going to create a shadow. If my hypothesis is correct, the shadow will be visible because it's being formed on the opposite side of the light, and that's where the light must be sensed. That's where the elements that receive the light and transduce the light must exist. And furthermore, if I move that candle back and forth, the shadow should move. If the candle goes up, the shadow should move down. And so he used that argument conceptually to actually measure the distance between the blood vessel and where the shadow is sensed, where the light is sensed. Now, this is not a new idea, actually. Astronomers had been using this kind of argument for a long time. They had used in order to measure the distance between stars and planets from the sun and from the Earth. So here's how the argument worked. Suppose you're on Earth in the springtime, and you're looking at some celestial object like a planet or maybe a nearby star, and you see it against a backdrop of fixed stars that are so far away that they don't move when the Earth moves. So now you make your measurements of where does that little shadow or where does that object exist relative to the fixed stars? It's right there. And now wait 6 months until the Earth gets on the other side. Instead of spring, it's now autumn. And you look again, and lo and behold, the object seems to be someplace else. It's moved. That's the parallax that's being used. And based on a little bit of geometry, you can calculate the distance between that object and the sun, or distance from the Earth, and make those measurements using in an astronomical context. But in the same way, Müller said, let's take that light, move the light, see how far these shadows move, and we'll calculate the distance between blood vessels and the shadows and where they're being detected. And it turned out to be about 0.2 of a millimeter. And he said, by golly, that's just about where the photoreceptors sit. So there's my proof. You see, that's his arguments. Now, although Müller's results were consistent with his hypothesis that vision is initiated by rods and cones, he actually did not claim, in print anyway, that his parallax arguments provided a definitive proof. Instead, his claim was more guarded. He was careful with his words. And here's a quotation from his paper, that famous 1854 paper. He said, "It would be inappropriate to conclude from the data whether cones or external grains, meaning which are the photoreceptor nuclei, might they be the sensitive elements? We don't know. But for one of those outer layers, the conclusion is inescapable, for the light is now perceived before it reaches the outer layers where the vascular shadows fall." So in short, he's saying, all I know for sure is that the light is being sensed on the other side, the outer side of the retina. And there are several candidates out there. I can't be absolutely certain it's the rods and the cones, even though I think they probably are. So Müller was basically being cautious. To implicate the rods and the cones would require elimination of the other elements, which were poorly understood. Nevertheless, this is an important part, subsequent authorities, they put words into Müller's mouth. People like Schultz, who wrote a definitive treatise in 1866, Helmholtz, the great Helmholtz, Poliak's treatise, everybody quotes Poliak in the 20th century, and also Rodiak. And most recently, Werner et al., this just recently, past year. They all attributed to Müller what he did not attribute to himself. They said that he proved it, that rods and cones are the recipient of light. Now, In that same year, 1854, Bergmann gave an equally compelling argument based on anatomical grounds. Unlike Müller, Bergmann had access to French human retina to examine. And at the center of the human macula, Bergmann noticed that the fovea area is not a hole like other people had thought and had stated in their work. He says it's simply thinner than the surrounding regions because all of the layers have vanished except the bacillary layer. And he says, in fact, he thinks they were displaced laterally because the layers of the retina are actually thicker immediately outside the fovea than they are in other peripheral areas of the retina. So you just imagine that those elements have been shifted laterally. Now, Bergmann reasoned that since these rods and cones are the only elements present in the fovea, they absolutely must be the light-sensitive elements of the retina. You can't get around it. And moreover, Bergmann observed Müller's fibers in the peripheral retina. Those are the ones spanning the retina. He said, yeah, I see them, but in the fovea, they're not oriented radially. They're oriented laterally, obliquely. And he said these oblique fibers have to be there in order to connect the receptors to the rest of the retina. And so they must run diagonally away from the center of the fovea, just as the modern data that I showed you illustrates it so beautifully. And so Bergmann suggested that these fibers are part of an anatomical pathway from fovea to cones, from the foveal cones and rods to their corresponding optic nerve fibers that have been displaced laterally from the fovea. Now, it'll turn out that Müller will bitterly dispute this claim, but that's what Bergmann said. So here are Bergmann's arguments in his own words. He said that the fovea centralis is located in the middle of the most acute part of the retina, is of course not a blind spot, which is what it would be if it was a hole. Rather, it's an unusually constructed part. It can be assumed to be particularly advantageous. In fact, it is only those retinal elements present here that can be assumed to be precipient, meaning responsible for perception. The assumption would therefore admittedly be very much in favor of the cones, as little speaks in favor of the granular layers given their structure. So he too says he's like Bergmann, like Müller. He said, I'm not absolutely positive that it can't be the granular layers, but in the end, it's going to turn out to be an insignificant point because the granular layers actually are part of the cones. Those are the nuclei of the cone photoreceptors. So it becomes a moot point in the end. Now, here's the sad part. Within a few years, Bergmann's work was completely forgotten, and his argument has been neglected by all subsequent scholars. Although cited in passing by later authorities, a modern recapitulation of Bergmann's argument does not appear to exist in the literature. I've looked, I cannot find it. That argument is that the rods and cones must be the light-sensitive elements of the retina because they are the only neural elements present in the fovea. So given the fundamental importance of identifying the light-sensitive elements of the retina, it seems to me surprising that Bergmann's work has been ignored by the very same authors who praised Müller and gave him more credit than he himself was willing to assume. So the question in my mind when I started this project several years ago was, why has Bergmann been ignored for 170 years? It just didn't make sense to me to be ignored for such a long time. And I think that the likely explanation is that Bergmann's 1854 paper offered only written reports, descriptions of his observations. He did not provide any drawings or any objective evidence of his anatomical observations. He simply described what he saw. That omission made it easy for his critic, Müller, who was a highly regarded anatomist, to cast doubt upon Bergmann's discovery. And he claimed in print that Bergmann's anatomical sections must have been damaged, Therefore, his observations were artifacts and his conclusions are invalid. It was a damning criticism. Bergmann was obviously and clearly disappointed by Müller's skepticism, and he actually sought to regain a little bit of acknowledgement for his accomplishments by publishing a second paper in 1857. It was actually a paper on a different topic, but the first part of the paper is a kind of rebuttal to Müller. And he showed in this one now the type of anatomical drawing that he failed to include in his original 1854 paper. He actually says, you know, I made a mistake. I should have included a drawing, something like this one before. My humblest apologies. This is the best I can offer. Now, he said, this is not my best data drawing because, unfortunately, over the past 3 years, most of my specimens have shriveled up and gone away due to poor fixation. But he had 1 or 2 that survived, and so he was able to make some drawings. And here's the best example he could come up with. This is the best he could offer, which unfortunately doesn't show the retina right in the center of the fovea, but it's nearby. It's at the foveal rim. And what you can see is the optic nerve fibers up here are thinning out. It's actually extra thick just outside the fovea. He said that's where they're piling up, and then it thins out very much, so there are no optic nerve fibers in the center. And likewise, the ganglion cell bodies pile up outside the fovea. It's extra thick here, and then it's going to thin out. And if I only had a section that went a little bit further along, you'd see they disappear completely, as do the inner plexiform and the internuclear layer. All of those disappeared. I said that in 1852, and I wish I had a picture I could show you now to prove it. This is the best I've got. Now, if you take a comparison, make a comparison between that figure from Bergmann, the best he had available, and a really definitive drawing of the same kind by Polyak, which is reproduced many, many times. Just about any textbook of retinal anatomy in the 20th century includes a picture from Polyak, one like this, and you'll see they're very similar. Thinning of the retina, thinning of the ganglion cell retina layers, thinning of the internuclear layers. And look at this, horizontally oriented or obliquely oriented fibers running from the cone and rod nuclei. Those are now known as the fibers of Henle. Bergmann showed the very same thing, but they're not called the fibers of Bergmann. They're called the fibers of Henle. Now, yeah, why did that happen? It seems very strange. I tried to track that down, and the best I could do is come up with a very nice and highly regarded treatise on retinal anatomy by the authority of the second half of the 19th century, Mr. Schultz. Wrote this book on the anatomy and physiology of the retina, another German writer. And he confirmed Bergmann's observations. He confirmed everything I've just been saying about thinning of the retinal layers and the obliquely oriented fibers, but he doesn't credit them to Bergmann. Schultz credited Henle, in fact, as having discovered a whole new layer that nobody knew about. These obliquely oriented or laterally running fibers, he said, Henle discovered those. Well, if you read Henle, Henle said, I didn't discover them, Bergmann did. Right? So here we have Schultz crediting Henle having discovered a new retinal layer of obliquely running axons from photoreceptors that had been reported by Bergmann, denied by Müller, and credited to Bergmann by Henle, but Schultz was the last one in the list of people, and he said, no, forget Bergmann, it's Henle. So forevermore, these fibers are known as Henle's fiber layer. So I can make some conclusions now. In that pivotal year of 1854, Müller and Bergmann independently presented convincing evidence that rods and cones are the light-sensitive elements of the vertebrate eye. Müller's argument was physiological, based on visual perception of shadows of blood vessels, but his measurements were not precise enough to specify which elements of the outer retina detected the shadows. But he was right, it was the rods and the cones. Bergmann's argument was histological, based on his observation that all other retinal layers were missing from the fovea except the bacillary layer of rods and cones. He too was right. So taken together, these independent, complementary arguments suggest that Müller and Bergmann really ought to be recognized as co-discoverers that rods and cones are the retinal photoreceptors responsible for initiating vision. Now, it turns out there are some missed opportunities. They could have been preceded by other people to make the very same discoveries. People like Bell, 1812, Steinbach, 1813, Purkinje himself, 1819, and Wheatstone, famous scientist, 1830. They published accounts of entoptic perception of blood vessels, but they didn't take the very next simple step of using that evidence to draw the obvious conclusion that the vessel shadows must be detected by light-sensitive elements in the outer half of the retina. In other words, the rods and the cones. They could have done that. Maybe they just thought it was so obvious, why bother? Who knows? But they could have been the discoverers. Meanwhile, Horner, 1834, he was an English scientist, and he actually performed Müller's exact parallax experiment 20 years earlier. And he got the same answer, that receptors are about 0.2 of a millimeter behind the blood vessels. Horner's work was completely overlooked, perhaps because he wrote in English, and all the great authorities of the day were writing in German. Perhaps the Germans didn't read the English literature, who knows? But there's a lesson there about publishing your work in a language that other people are going to understand, perhaps. And Purkinje, Wheatstone, and Baumann all observed that the fovea is avascular, no blood vessels there, but they failed to make the inference that the inner layers of the retina where blood vessels are known to exist, they probably are missing. Such an inference would have preceded Bergmann's conclusion that only rods and cones exist in the fovea, so they must be the photoreceptors. So, in the end, it's Bergmann and Müller get the credit. Now, my story's running on a bit late here, but I got to tell you, it's not the end of the story, because there were lingering doubts, important doubts that persisted for more than a century. Not everybody was convinced that rods and cones were the. And now the main reason, the biggest objection, is the issue of visual pigment. Where is the visual pigment that absorbs the light? You see, every retinal anatomist prior to 1854 commented on the amazing transparency of the retina. It's completely transparent, including the rods and cones. In fact, Müller and Bergmann both gave the teleological argument that transparency is an essential property of the retina because it enables light to pass through the thickness of the retina to the rods and the cones without being absorbed. But according to that argument, light should pass through the transparent rods and cones as well, without any effect. And the reason that is an important argument is because it was already known, the law of photochemistry established by Grotius and Draper, that light must be absorbed to have an effect on matter. But at that time, it was really a very confused time. Nobody knew the means by which light interacts with retinal tissue to elicit a physiological response. It was a great mystery. Even the great Helmholtz commented as late as 1867. He said, we have no idea how light affects cones, whether a vibration is set up, whether there's a molecular change, whether there's some heating effect, or whether this light-sensitive layer of the retina is part of some photochemical apparatus. We don't know. Well, that central mystery led a young German scientist by the name of Franz Boll to investigate retinal pigment epithelium as a possible alternative to rods and cones, because the pigment epithelium is in exactly the same place as the rods and the cones. They're right next to each other. So maybe those are the photoreceptors, because they have pigment. They can catch the light and turn it into a neural signal. So he imagined that the pigment of the retinal epithelium might be the visual counterpart to chlorophyll, which of course is the pigment responsible for photosynthesis in plants. Maybe that's where the visual pigment lies, in the epithelium. And when testing his hypothesis experimentally, he accidentally or inadvertently discovered rhodopsin, the photolabile visual pigment that exists in rods. And he did it while dissecting the eye of a frog. Bull described his accidental or serendipitous event as follows. He said, if one bisects an eyeball and with very fine tweezers pulls the retina away from the dark background of the pigment epithelium and choroid, the retina looks intensely red in the first moment. But during the next 10 or at most 20 seconds, this color fades gradually until it disappears completely. Isn't that startling? Well, that solved the mystery, you see. Bull's discovery of the rapid bleaching by sunlight of rod pigment explained why anatomists had long failed to realize that rods and cones are potentially photoreceptors. They were doing their dissections under the strong sunlight coming through a window, and the very process of dissecting the eye under that strong illumination would have bleached the visual pigments. So by the time they went to look, they were transparent. But inside the closed living eye, said Bohl, the rods aren't transparent. They contain a visual pigment, which he called erythropsin. That is impossible to pronounce. Fortunately, his contemporary Kuhn renamed it rhodopsin, and this pigment was found to bleach when it absorbs light, and the bleaching process uses the energy of the light to initiate a neural response. Well, the discovery of rhodopsin by Bohl and the subsequent elucidation of his chemistry by Kuhn rescued Bergmann and Müller's claim from the objection that rods and cones transparency is actually incompatible with them being photoreceptors. But to tell you the truth, it took another century for their claim to be universally accepted. There were still lingering doubts amongst 20th century electrophysiologists using intracellular recording from rods and cones, but they were able to demonstrate unequivocally that rods and cones are inherently photosensitive, thereby eliminating any possibility of neural contribution from other retinal cells or the pigmented epithelium. There was, in fact, in that time, and now actually in the third millennium here, a very unexpected surprise. Remember that early 19th century hypothesis that retinal ganglion cells are light sensors? Well, guess what? It actually proved to be true for a very small population of ganglion cells containing the light-sensitive pigment called melanopsin. However, the interretinal location of those ganglion cells suggests they are not involved in visual perception. They're thought to mediate photoreceptive tasks like the regulation of circadian rhythms and the acute suppression of pineal melatonin. Okay, let me sum up. In 1854, Bergmann and Müller independently reported evidence confirming earlier speculations that rods and cones are the light-sensitive elements of the retina. Their arguments were convincing, but not immediately conclusive. The transparency of rods and cones appeared to discount them initially as being light receptors, but that objection was overcome by the subsequent discovery of visual pigments in those cells. Difficulties also appeared from Müller's conception of functional connectivity from photoreceptors cells directly to optic nerve fibers, but they were soon resolved by Schultz's investigation confirming Bergmann's interpretation of the obliquely oriented fibers of Henle and presaging Cahal's neuron doctrine. So in short, Bergmann and Müller hypothesis that rods and cones are photoreceptors has withstood the test of time. Bergmann's unique contribution has been long overlooked, but the documentation and historical analysis presented today show that he should be recognized as a co-discoverer of the anatomical site in the retina where vision is initiated. The advances reported by Bergmann and Müller in that pivotal year of 1854 are important milestones in vision science, that eventually led to the modern concept of rods and cones as specialized neurons that initiate the visual process. Henle's fibers, or the photoreceptor axons, transmit visual signals generated by light to the bipolar interneurons of the internuclear layer, and in turn, those interneurons relay their visual signals to ganglion cells and their axons, thus completing the three-neuron chain from the retinal image to the retinal neural representation transmitted by the optic nerve to the brain. Many scientists contributed to these advances in knowledge, and everyone deserves recognition. Now I'll finish with a brief epilogue, the moral of my tale. The 100th anniversary of Bohl's discovery of rhodopsin, which stimulated so much work by Kuhn, was celebrated. By the way, Kuhn is famous and Bohl wasn't. Same story all over again. That was celebrated by the publication of English translations of their work in a feature issue of the journal Vision Research back in 1977. And an introductory editorial to that feature issue by Frederick Crescitelli emphasized the great importance of their work and that of other forgotten scholars in solving the mystery of light sensitivity by rods and cones. And this is what he had to say. In this issue of Vision Research, we commemorate and revitalize the labors of great investigators who are already unknown or forgotten by many living physiologists. It is an old and repeated tragedy that living man tends to forget or ignore the past, often to his detriment. So this issue of Vision Research may help rekindle interest in the history of visual science in which they were engaged. It's also the history of a period in which science, as we know it and enjoy it today, rose to eminence and took its place as one of man's most noble activities. And so for these reasons, I'd really like to congratulate the Spanish Optical Society for initiating the history of optics, this history of optics group, especially Sergio Barbero, who began this group, because they are able to rekindle, hopefully, interest in optics in all its forms, including visual optics. And I believe that the history of visual optics is especially important to preserve because visual optics transforms the traditional branches of geometrical and physical and quantum optics, which operate in the theoretical and physical domains, into the domain of the human reality and the eye and visual perception. And it's through this transformation that the science of visual optics makes its greatest contribution to mankind and our collective history, because we are the ultimate consumers of images. Our eyes and our brains give meaning to light. So thank you very much for your kind attention. I'll remind you once again that the presentation, at least the novel parts of this presentation, have been published in the Journal of History of Neuroscience. And if anybody would like a copy of that reprint, simply send your request along and I'll be happy to fulfill it. Thank you very much.